Hello everyone. Let's talk about identification of genes on the chromosome. Oftentimes when you read a textbook, you come across information which says a particular gene is located on a particular chromosome. But the question is, how do scientists know which gene is located where? The dyes made up of different organic compounds were widely used for the staining of chromosome. This allowed scientists to study mitosis and meiosis. By 1970s, scientists were using dyes that stain the chromosome into dark and light bands. This technique was known as chromosome banding. We all know a cell can have many chromosomes. Using a technique known as pulsed field gel electrophoresis, the chromosomes can be separated. Using pulsed field gel electrophoresis, the chromosomes can be isolated from normal cells and the abnormal cells. After all chromosomes are separated, the chromosome of interest is purified from the gel. If you observe this carefully, the abnormal cells will have a defective chromosome in which one band is missing. The same chromosome isolated from the normal cell will show presence of that particular band. After chromosomes are isolated, they are treated with a restriction enzyme. The digestion products are then analyzed by agarose gel electrophoresis. Since the abnormal chromosome has the absence of a particular band, the DNA gel will also demonstrate absence of DNA band. The DNA gel may show absence of several bands. However, to keep things simple, here we have shown absence of only one band. If we compare the digestion products of abnormal chromosome and the normal chromosome side by side, then the normal chromosome will show presence of certain bands that were absent in the abnormal one. And this is the band that we are interested in. So the next step is to purify this band from the gel. This is then ligated into a plasmid. This plasmid is finally transformed into the host where it is maintained. The plasmid carrying this insert can be isolated from the host anytime when required. The insert in the plasmid can be isolated using RE digestion and sent for DNA sequencing. The DNA sequence obtained will have the gene that was absent in the abnormal one. Now, besides DNA sequencing of insert, there is also a second method. In this method, mRNA is first isolated from the normal cells. This mRNA is then converted into cDNA. And cDNA is used to prepare cDNA library. The cDNA library will have information of all the genes that were expressed in the normal cells. Just like plasmid is maintained in the host, the cDNA ligated into the plasmid is also maintained in the host. Each host will grow and form a colony. Once colonies are grown, they are transferred on the membrane. Once colonies are transferred on the membrane, the colonies on membrane can be lysed using a detergent and sodium hydroxide. The presence of sodium hydroxide will also denature double-stranded DNA into single-stranded DNA. Once this is done, we isolate the plasmid from the previous host. If you can recall, this plasmid has the chromosomal fragment that was present in the normal cell but absent in the abnormal one. This chromosomal fragment in the plasmid can be labeled with P32 and used as a probe. So we will remove this insert, label it with P32 and use this as a probe to hybridize with the DNA present on the membrane which contain the cDNA library. The colony that shows positive binding with the probe can be traced back to the original plate. The plasmid from this colony will have cDNA that was complementary to the probe. Hence, if you observe carefully, then the cDNA present in the plasmid is the gene that was originally present on the normal chromosome but absent in the abnormal one. So, to summarize, we first isolate the band that was present in the normal chromosome but absent in the abnormal one. This band is ligated into the plasmid and used as a probe to identify the cDNA. The cDNA represents the gene that was absent in the abnormal chromosome but present in the normal one. Since cDNA is maintained in the plasmid, the protein product made by the cDNA can be purified and used for further studies. So, this is how the genes present on the chromosome are identified.